together mobilizing bioacoustic data to meet conservation goals. I'm here joined by Talia speaker from Wildlabs based at WWF US. Well, my name is Vanessa Reyes, also from Wildlabs based at WCS Argentina. And also Justin Kitsis from the University of Pittsburgh from USA, who is joining us virtually, so he will be interactive through the chat, through the Zoom. So this session follows a collaborative effort led by Wildlabs to identify key innovations and priorities that could shape the next decade of bioacoustics research and applications. So Wildlabs is the world's largest conservation technology community with over 13,000 members in over 170 countries. It's a space where conservation practitioners, researchers, and technologists come together to share ideas, find collaborators, and find useful resources for the conservation projects. So at the heart of effective conservation is our ability to measure what is happening in nature. And as we've been hearing through this conference, that depends on how we collect data and process it and manage the data and use the data. So over the past decade, technology has really transformed the way we collect information about species and ecosystems. We now have a different range of sensors to help us collect information. AI also helps us identify and detect species. And we have platforms like MoveBank and Wildlife Insights that help us turn huge data streams into actionable insights. So combining these innovations with advances in ecological methods and monitoring methods will help us reach global conservation goals like city by city more efficiently. And among these technologies is bioacoustics. But for bioacoustics to reach its full potential, that pieces in this data pipeline needs to connect seamlessly. And so how can bioacoustics help us transform our understanding of life on Earth? And the sounds made by wild animals provide us a powerful way to understand nature during a time of a global biodiversity crisis. And passive acoustic monitoring, which is the use of acoustic recorders to collect sounds over large areas and long periods of time, supports many research and conservation applications. It allows us to study species, populations, communities through sounds. Bioacoustics can now help us also tackle ocean challenges, like from detecting wildlife crime and assessing environmental impacts to supporting nature restoration and nature-based economies. But yet, despite all this progress, we are at a turning point where technology is advancing quickly. We have sensors which are getting cheaper, AI is stronger, and acoustic data is growing fast, but progress remains uneven. And many regions, especially those with high biodiversity, still lack access to data and resources. So what is needed are shared priorities and target support to help a global community work together and turn innovation into real impact. And so that is why with Wildlabs, we conducted Bioacoustic Horizon Scan, which was a global participatory process supported by Gordon and Betty Moore Foundation and guided by an extraordinary advisory committee and a global experts panel. The goal of this effort was to identify opportunities and challenges that could shape the future of bioacoustics and to build a shared roadmap that guide research, innovation, and funding. And so horizon scanning provides a structured foresight method to identify priority areas, assess their feasibility, and estimate timeframes and resources needed. So we follow a 5-step approach where topic sourcing involves getting ideas from the community, on what they believe could represent key challenges and opportunities to shape the future of this field. And topics could span short-term challenges addressable through significant resource mobilization to more speculative blue sky ideas that will require long-term innovation in alignment with the Three Horizons framework. Once we collected these topics through an open online form that was widely distributed across different networks, we consolidated the topics into a list of 63 distinct topics, which were then prioritized by a group of 50 global expert panel. And then we have facilitated workshops to discuss this short list of priorities. And after that, the expert panel rescored the refined list and come out with 20 final technology-related priorities. And these 20 priorities, we group them into 5 broad outcome-oriented domains, which are, one of these areas are next generation recorders. There is a need for more affordable recorders with high quality sound and features like sound localization and real-time edge processing, enabling rapid detection and human intervention to critical events. The second priority area refers to new analytical methods, especially in relation to novel approaches of AI. And AI is reshaping analysis for species detection and soundscape interpretation, but models should be ground trusted with independent data and aligned with clear ecological goals to guide meaningful conservation action. So also within this domain, improved abundance estimation methods was identified as a priority, and also automated individual identification to enable tracking and assessment of behaviors and demographic studies through focal fingerprints of individually distinct sounds. The third priority is global and inclusive participation. So for bioacoustics to really reach its full potential, it requires sustained investment in capacity building, inclusive infrastructure, accessible tools, and funding strategies that support local engagement and data sovereignty. The fourth domain we identify is streamlined data exchange and integration, which means managing the increasing volume of acoustic data through coordinated solutions for storage, interoperability, and accessibility. And finally, scale applications and impacts. Bioacoustics is increasingly used in environmental impact assessment and biodiversity frameworks, but implementation remains uneven. So to scale impact, we need clear communication, simple metrics, and accessible software that link science, policy, and practice. So in summary, our Horizon Scan identified these 5 connected areas that can shape the next era of bioacoustics. Progress in one area drive progress in others. So this creates a connected system that can move bioacoustics from isolated projects to coordinated global monitoring. These priorities represent a shared vision of how bioacoustics can meet 3 challenges of biodiversity loss and global inequities in scientific capacity. And by aligning these priorities with the Montreal Global Biodiversity Framework and Sustainable Development Goals, we aim to ensure that this work supports the global agenda to protect and restore nature. So today we hear from 7 talks that will explore this domain in practice. For time reason, I won't mention all of these right now, but each of them will highlight practical examples of these innovations in action. And then we will have some time for questions and discussions. Okay, Looking ahead, one of the most exciting opportunities that for us to continue these conversations is the next International Conservation Technology Conference that will be hosted by Wildlife in Lima, Peru, next year in February, with various partner organizations. So we hope we can see some of you there. And this event will bring together tools, people, and ideas from across the world to advance conservation technology. And we hope it will provide a space to continue building on the bioacoustic horizon scan priorities and turning these shared ideas into collaboration and real progress. And you can find more some cards with information on the conference and Wildlabs over there. And yeah, if you are interested to getting involved, you can also reach out to me or Talia. So thank you so much for your attention. I invite you all to join the discussion after the talks and to connect with Wildlabs. Thank you. So now our first speaker is Gabriel Chiari from Center for Data Science and Artificial Intelligence at Dedan Kimathi University of Technology in Kenya. And he will be presenting Pi while optimizing the Raspberry Pi for acoustic monitoring of ecosystems. Thank you.
Good afternoon. So my name is Gabriel Chiari. I'm from Kenya, and we'll be talking with you about PiWild, which is a system for optimizing the Raspberry Pi for acoustic monitoring of ecosystems. So this was work that I did when I was in my former role as a research intern at the Center for Data Science and Artificial Intelligence. That's at Dedan Kimathi University of Technology, Kenya, before I moved to my new role as a bioacoustics research fellow at Natural States, still in Kenya. So most of the work, I mean, they are in the stage, the work is in the stage that was initially when I left, when I submitted this abstract. So an introduction has been made about bioacoustic, and so I don't need to go into details, but mainly what bioacoustic entails is recording various taxa using a recording unit, could be an autonomous recording unit or handheld recording unit, and then extracting features from the data, mainly the frequency component, that are passed to machine learning models for analysis. And then we can get conservation insights such as the biodiversity, occupancy, population, and all that. So there is a critical scarcity of biodiversity data in the global south, and especially Africa. And Africa hosts a significant proportion of animals that we have on this planet. And this can be attributed to lack of access to bioacoustic tools and limited technical capacity. So on the right, we see, for example, a study that was carried on eBird hotspots where people record sounds, and you can see there's less activity on the continent of Africa compared to the global north. So, and you get that tools such as Barnet are less effective on African bird species because they have been trained or exposed to more species from the global north. And this is something that we experience when we are using something like Barnet to process the data that we collect. So, and this severely hampers conservation in one of the most biodiverse continents on this planet. So to solve this, we started a project to collect audio data locally. And when we were starting this, it was just after COVID-19. And we didn't have access to things like audio mode because there were shipping constraints, restricted movement and all that. So, but we had some Raspberry Pis that were lying around. So we decided to modify them or to make them suitable to be deployed in the field. So the Raspberry Pi is just like a computer. It's a single board computer. And therefore, unlike microcontrollers, they may be a bit complicated when it comes to deploying them. It will be just like deploying your laptop in the field, and you may lack some functionality, flexibility, like scheduling the time you want it to record and then expect it to wake up later. So a Raspberry Pi cannot do that way. It doesn't have sleep modes, and that would, since it's a power hungry system, it would not be suitable to deploy it as it is. So what we did is we developed a power board to enable us to deploy the system autonomously in the field. And this is it. So with this system, we're able to deploy the system, and it can be able to record sound autonomously. We can be able to schedule the recording period, for example, early in the morning when we expect a lot of acoustic activity, and later in the afternoon. We're able to monitor battery voltage and allow for smart shutdown in case, for example, the battery gets drained and all that. So the Raspberry Pi has an edge compared to microcontrollers since it has a very high processing capability. For example, the Raspberry Pi 4 has a RAM of up to 8 GB. So with that, you can do some complex edge processing compared to what you would with microcontrollers. So, and the system is battery and solar powered. So the picture on the right is a deployment of the system in the university's conservancy. So the features of the system include so active streaming and then smart recording. So we are able to stream continuously, but save segments that have acoustic activity. So we tested LoRa inference transmission. So that's after processing the audio that we are collecting. But we are yet to test that in the field because we need to get our gateway on a place that it can broadcast on a wider area. So we have that aspect of edge AI. So with audio data, sometimes if, for example, you are streaming continuously and saving the raw data in .wav form, that may result to a huge space requirement and also power consumption when you're writing to the SD card. But with Edge AI, you can be able to save segments, for example, that have acoustic activity after maybe doing no bad audio classification. You could as well compress the data, for example, to lossless, .flag, or other lossy formats, saving on space. So the system also monitors battery voltage and therefore enabling it to shut down, for example, when the battery gets drained and wake up when it gets recharged. So I've talked about sound resampling. So then the autonomous control and scheduling, you can schedule your system to operate when you expect acoustic activities, then shut down during when you don't expect a lot of activities. Therefore, you save on energy and storage. So here is a sample deployment of the system. This is the Dedan Kimathi University of Technology Wildlife Conservancy. So we have deployed the systems in those squares that have the blue dots. That the squares are 200 by 200 meters. And here are some of the results from the system. So we deployed the system with a basic model. It wasn't the best, it was just to proof of concept. So it was classifying 3 classes, I think, of 3, yeah. So 2 bird species and the other class. So on the top there is a spectrogram of sample data audio data that was collected. And then the output after classifying the recorded sounds was saved in CSV. These are what we would opt to transmit if you want to do real-time monitoring rather than transmitting the raw audio, because that requires huge bandwidth and more power consumption. And then also here is a plot of the battery voltage profile. So we didn't expect the system to perform this well. You can see that the system wakes up at 11 a.m. in the morning. And then you can see that there's a downward trend up to when the sun comes up, and then the battery starts charging, and then you can see an upward trend. And you find that at the end of the deployment, the battery was almost full, and therefore means that we were not utilizing our system optimally. We could have, for example, extended the recording later in the afternoon, and we would have sufficient power for the system to run on. So there are some improvements that need to be done. For example, we need to get a good, better quality microphone, because we noticed the microphone that we're using was less sensitive. Yeah, and things to also employing LoRa, okay, on in the field. So that's the end of my presentation.
There is Medicine 10, yeah, from the Université de Montréal, Canada. And so we didn't have to talk about individual identification and also classification is quite slow. Hi, everyone. Super nice to be here. So I'm Mélisande. I'm a PhD candidate at Université de Montréal and MIVA Quebec AI Institute. And today I'm going to present some work that we've been doing on developing a method for identifying birdsong syllables without labeled data. So there's one big bottleneck in bioacoustics right now, which is that we now have access to a lot of recordings, but it also means that we have to spend more time annotating them, and it's really time and cost intensive. So there are many things that people in bioacoustics, I think, are interested to annotate in their recordings, but we took the approach of looking at the problem of syllable level annotations, because it's quite useful for many applications, such as like sensory-motor learning, or modeling the grammar and syntax of birdsong. So there have been like recent successes in machine learning methods for bioacoustics, especially for species classification. There's also a model that's called TweetyNet that also tried to do syllable level annotation, but those models are supervised. So it means that they rely on a certain amount of labeled data. Now, so to alleviate like this bottleneck of having to, in any case, annotate some data, we thought, okay, can we develop a fully unsupervised method for syllable annotation that might not be perfect, but it's still easier for experts to validate the annotations than actually delineate each syllable. So the approach that we took is as follows. So basically the goal is that if you have a bunch of recordings from like individuals from a given species, you want to try and extract a template that corresponds to one template for like each of the syllables that are represented in your record. So you first transform like your audio into a spectrogram, however you want to, and however it fits best to your species. And the first thing that we do is like segment syllables based on silences, which is like a usual approach that we do. So then what we do with these segments is we do a first clustering. So we apply like principal cone analysis and then run HTV scan, which is an unsupervised clustering method that allows to reject some syllables and for which you don't have to input the number of clusters that you need to find. So of course, this first clustering is not perfect, but it's okay. Like all you want to do is not have the perfect clustering of everything. You just want to find like the groups that are represented in your data.
And so after we do that, you can refine further your cluster. So we do a first step that is like that splits the clusters further and then merges them back together. I mean, I can provide more details, but the idea is that you can do that iteratively. And at some point, you end with a clustering where ideally all the syllables like within one cluster correspond to, sorry, all the events in one cluster correspond to the same syllable. In this approach, it's not so much a problem if you have too many clusters, because you can imagine that experts would just, say those are the same. It's just the same label. So then we can get templates from these clusters as the cluster medians. And then once you have these templates, you can apply matching pursuit on any recording, whether you've seen it or not. And so then you can obtain like the annotation of your recording. So we apply this to two different data sets. So the first one is we wanted to look into whether this kind of approach could help like for individual identification. And so we took a data set of Bengali finches that has human annotations to which we can compare our model performance to. So actually, I showed an imperfect example. So the top is like the human annotations, the bottom is like what our model produces. So you can see that it misses some syllables, but again, like the goal is not to have something, like we know that people are not necessarily going to trust something that is fully unsupervised, but it's quite easy for someone to say, oh, there's a syllable missing. I'm just going to add that to the template, and then you run the template match. And so what is quite interesting with this kind of approach is that usually annotations are not really consistent between individuals. So basically syllable A for individual 1 might not be the same as syllable A for individual B. And here what we can do with this approach is like really maybe study intra and inter variability between the syllables, sorry. And so here, like I show like for the 4 individuals in our data set, like what is the relative template occurrence per individuals for each of them. And so you see that they actually do have like different patterns in like what syllables occur. And some individuals, have some syllables that are very distinctive for them. So we think like that this approach could, be useful for this kind of application. And then the other thing that we looked into was whether we could also do other tasks like song type clustering. And so we took a data set of great hits. It's like the Witham great hits data set. And we took a subsample of like 25 individuals and 2,000 songs out of them. And there are like 71 like song types per times individuals that are represented. And so here it's just like a 2D representation of what the embeddings that we can obtain from just like the syllables that we identify with our method are versus like Perch embeddings, which of course it's not a completely fair comparison because Perch was trained like for species classification. But what we do see is that with our representation of bag of syllables, we're able to like see patterns of like clusters of song types more clearly than what we obtained with Perch. So I think that's it for my presentation. We have printed out. Right now, we're looking for people to collaborate with. If like people have data sets that they want to, I don't know, and they have interesting questions where they think like our method could be useful for, we're very happy to hear about that.
So we'll take all the questions at the end. So, okay. So our next speaker is Mao Ning Tuamu from the Biodiversity Research Center, Academia Sinica, Taiwan, who will be presenting enabling large-scale acoustic biodiversity data mobilization through open and automated data flows.
Okay, good afternoon, everyone. I'm Mao Tuamu from Academia Sinica in Taiwan. So today I would like to share how my team developed a semi-automated data flow to speed up the process from the field recordings to open and ready to use biodiversity data. So, okay, yeah, so to track the environmental and biodiversity changes across Asia, we developed an Asian soundscape monitoring network through collaborations with both domestic and international partners. This network so far has collected over 23 million minutes of recordings from a wide range of habitats. So the recordings and also the derived biodiversity information have a huge potential for improving our understanding of how biodiversity responds and adapts to environmental changes. However, handling such massive and unstructured audio data and extracting meaningful biodiversity information is time consuming, and it can often delay the data applications. So now let me walk you through our data flow, which I believe is typical for most of the passive acoustic monitoring. So we start by collecting sounds in the field using autonomous recording units. After retrieving the memory cards, we checked for damaged or missing files. We standardized the file names. We created the metadata. The recordings are then stored and backed up before we can analyze them to extract biodiversity information. And usually, we also need to validate the information, especially for that generated by AI. And finally, both the recordings and the derived data are published in a data portal for others to use. So you can imagine almost every step in this process involves a lot of manual work. So to make this more efficient, we developed a tool interface that automates several steps in this process. For example, it can automatically detect incomplete or missing recordings based on the file size or the recording schedules. It can also display the spectrograms so users can quickly identify the potential data issues. It can also directly, automatically stores and backs up the recordings in designated folders. And once the new recordings are uploaded to our system, a series of routine analyses are triggered. These include generating long-term spectrograms, calculating several acoustic indices, detecting human voices, and using two AI-based approach to identify species vocalizations. So we built a dashboard to track the processing progress. So for each site and each month, the indicator light show which analyses are complete. To support the expert validation of the AI-generated species identifications, we built another interface which lets the experts easily search for the specific detections, play the audio clips, view the spectrograms, and finally annotate the results. So both the AI identifications and the experts annotations are stored in our databases and linked to the original recordings, helping us to evaluate and also continuously improve the AI performance. And our data portal provides an easy way to search, explore, and also access both recordings and the derived data. So for each download of the data, each is assigned an ARK identifier, similar to a DOI, providing an easy citation and tracking of the data usage. So to protect privacy, the recordings containing human voices cannot be directly accessed. However, their derived data remain available. So far, our system streamlines the data processing, cleaning, and also the data storage. It automates the information extractions, facilitate the AI validation, and also provide a, and also provide a user-friendly data portal for sharing and reuse. And okay, thanks. And yeah, and so this significantly reduced the need for the manual work. Our next steps are to adapt common data standards to make the integration with the other data sets easier, and to explore the wireless data transmission for the near real-time applications in biodiversity monitoring. So with that, I would like to thank my amazing teams, our funding agencies, and all collaborators for their support. And thank you for your listening. And I warmly invite you to explore the Asian soundscape on our data portal. Thank you.
Our next speaker is Nicolás Betancourt from the Instituto de Recursos Biológicos Alexander von Humboldt, Colombia, who will be presenting about palm flow analysis and standardization for passive acoustic monitoring data.
Works. Hello, I'm Nicolas, and today I'm here representing a part of what is the Humboldt Institute acoustic groups to show you a computational development for processing acoustic data and particularly passive acoustic data coming from money. Throughout different projects across the country, we have faced and identified what we believe are the main challenges preventing passive acoustic monitoring from achieving its full potential. The first of them is the lack of a common adopted standard, facilitating the sharing of data and collaborating with others. Also, the scarce integration between the existing open source computational tools, forcing teams to devote time of their projects to computational development. And those tools that are integrated, they are not commonly open source, and that's particularly troubling to us because we believe that that prevents accessibility, as it often relies on paid licenses. It also difficults reproducibility, as non-open code acts as a black box, and also it difficults to tailor workflows to particular research needs. Under the light of these challenges, thank you very much for your attention. A lot of spoilers. There you go. Under the light of these challenges, we have developed PalmFlow, which is a Python data pipeline for acoustic data. It covers what we believe are the main stages of most of passive acoustic monitoring projects. The first of them is translating field collected data into standards convenient for later stages of the process. Afterwards, we proceed with a quality control of the data in order to avoid wasting valuable computational resources on faulty data, and also to keep statistical conclusions on later stages clean. And finally, the most exciting part is the soundscape characterization, which include, among others, species detection. All of this is done under the light of existing data standards from which you will hear later on other presentations. So in such a way that outputs of PAMFlow are can be immediately uploaded to international and global repositories such as GV. We hope that this will increase the number of biodiversity records coming from passive acoustic monitoring. And another nice thing about PAMFlow is that it has a hybrid nature, because even though these three main stages can be run exclusively through command line prompts, thus not requiring code training, it is designed in such a way that you can tailor it to particular research needs, because it is a modular design. PAMFlow's outputs are not exclusively standardized CSV files, but you can, oh my God. You will also have visual aids for making easier the process of checking, for instance, in the number of recordings. In this graphic, you can visualize the number of recordings per sensor at a specific date, so you can easily detect malfunctioning sensors. In this other one, you have a deploy of the geographical location of your sensors, as well as summary metrics of your survey effort. In this final example, you have summary metrics of the later stages of the process, such as the species detection. And what is remarkable about all of these visual examples is that they come from actual projects that we have undertaken in the past. And this was a large project that we processed, in which we processed on average 100 hours of recording in just 1 hour, using the capacity of less than a half regular laptop. Accessibility and efficiency is crucial to us, because we want this tool to be used by rural and indigenous communities, which often don't have access to large computational capacity. Actually, we're going to use it in a project called Monitor Amazonia here in Colombia, where the rural people will actually lead the project of collecting the acoustic data and processing the data themselves. And now I'm finished. Thank you very much.
Our next speaker is Sam Govart from Research Institute for Nature and Forests in Belgium. And she will talk about, can we use the camera trap data package standard for bioacoustic data?
Hey, hello everybody. My name is Sanne Govaerts, and together with my colleague Peter de Smet, I work for the Research Institute for Nature and Forest in Belgium. And Julia Wil and Benjamin Crotois from the Norwegian Institute for Nature Research, NINA, in Norway, they came to us with a question if we could extend the camera trap data package standard for bioacoustics data. And then at the same moment, people from Humboldt also came with the same idea. So that was pretty cool. So spoiler alert, yes, we can. So we already had a workshop this morning. So for the people who joined that, sorry, it's going to be a little bit well, it's all going to be a repetition. I will guide you through the process, what we did. I will also share our key suggestions of how we can extend ChemTrapDP, and also I will explain how you can contribute in the development of this data standards. So we got funding from the Boring Fund to start a little project called Safe and Sound. And we reviewed existing standards and guidelines for bioacoustics. We reached out for users and collected use cases, but this was mainly in the networks that we already have. So it's good that we now can reach a more diverse public. So I need to point there for me. Yeah. The conclusion of that project is that, that we think there is a need for a widely accepted bioacoustics data standard that is not too complex and that can be used for both terrestrial and marine habitats. And we think that camera trap data package is a good fit because camera trap data has a very similar structure. And we also think that we can extend this standard so it can be used for both acoustic and camera trap data. So short ChemTrapDP, long camera trap data package. I will, oh, I have the same. Close your eyes. It doesn't work very well. Oh, where do you need to point? Ah, okay, yeah. Ah, okay. Well, so the original need to develop ChemTrapDP was because there was a need to have a data exchange format for camera trap data. So this is a little screenshot of the website, and there you can find all the detailed information of the metadata, the data, an example data set, frequently asked questions, and also the GitHub repository where the data standard actually lives. Bubniki et al. They wrote a paper that gives you a very good introduction to the data standards. ChemTrapDP was community developed and is actively maintained. And it follows the FAIR principles, meaning that data should be findable, accessible, interpretable, and usable, reusable. So there's a kind of ecosystem of tools emerging around ChemTrapDP that allows you to read, visualize, analyze data in ChemTrapDP formats. And there's also data management systems that adopted ChemTrapDP or are planning to do so. I'll wait until you have your pictures. Yeah. And also since December, ChemTrapDP is also a GBIF publication format, means that you can just upload ChemTrapDP to the IPT and there's an automatic transformation happening to Darwin Core and EML because that's the format that is displayed on GBIF. But it's still possible at the dataset level to download the original and so more rich datasets. So what does it look like? ChemTrapDP is based on another data standard called frictionless data package. It's the same one that also Darwin Core data package makes use of. And it consists of 4 files, data package.json, and 3 tabular files, the deployments, media, and observations. So I'm gonna guide you through this figure from the paper. At the highest level, we have metadata, the title, the co-authors, the license, this kind of stuff goes into data package.json. Then you have specific information about your placement in the field. So as soon as you start recording until you stop recording, or maybe emptying the memory card. And you have a setup, you have coordinates. This kind of information goes into the deployments table. Each deployment has a deployment ID. Then for each deployment, you will probably have 1 or more media files. They are linked by deployment ID. They also have specific information such as the timestamp, the file path, the capture method, and so on. And they're linked by deployment ID. And then last, ooh. Oh, yeah. Am I doing something? I'm not touching anything. Yeah. So based on the observation, you can, based on the media files, you can derive observations, which can indicate if you recorded or annotated an animal, just a blank, and the scientific name, the sound, the count, the life stage. Also, each observation has an observation ID, and its link can be linked by media ID and deployment ID. Oh, this is a bit annoying. Okay, so for ChemTrapDP to work for bioacoustics, we need to implement some changes. So this is just a very short introduction to what we're planning to do, or what we're proposing at least. First of all, the names need to be neutral and not camera specific anymore. Second, we need to add terms that are acoustic specific. And we also need to extend descriptions or maybe even add tables. So for the next, oh my God. For the next steps, we want to continue the discussion on GitHub. So at the bottom is the link, Tadwig/ChemTrapDP. Each suggestion will be posted as an issue on the GitHub. We also need to do some more testing, so to make sure it's not only theory, but it actually works in practice. And then if we get to consensus, we can prepare a new version of ChemTrapDP. Okay. So how can you be involved? You can watch the GitHub repository. You need to have an account and click the watch button. It's to get notifications. Second, you can also actively contribute to the issues. So provide your comments. Would be very useful with use cases. And second, it's also useful if people test their data. The links are a different repository. And we're doing the same parallel process also for insect AI data at this point, because the current standard was more focused on mammal researchers. So thank you very much.
Yeah.
Yeah.
So please remember to keep your questions until the end of the session. We're going to change the pointer. But our next speaker is Ellie Knight from University of Alberta, Canada, who will be presenting mobilizing bioacoustic data with wild tracks.
All right, thanks everyone for coming today. So today I'm going to be presenting an online software application called WildTracks for the storage, management, processing, and sharing of remote sensing data, including bioacoustic data. WildTracks is produced by a whole team of folks, of which I am not really one, at the Alberta Biodiversity Monitoring Institute, and supported by the University of Alberta and Biodiversity Pathways in Canada. And because WildTracks is available in both French and English with built-in language support, I'll provide the title in French today, La mobilisation des données bioacoustiques dans WildTracks. So we all know that passive acoustic monitoring is collecting vast amounts of data these days, enabling us to ask new and exciting questions about acoustic wildlife and phenomena. But those data are made most valuable when they're supported by robust data systems for managing and sharing data and standardized metadata. And it's that knowledge sharing and data security that are really important for building trust and collaboration and really making the most of these huge data sets that are being collected. And so WildTracks offers an end-to-end solution for that data management and storage, annotation and verification, and publication and sharing. And I'm going to go through each of these three sets of tools in the talk today. So the first is data management and storage. When you upload recordings to the WildTracks interface, they're stored and owned at the organization level, with recordings annotated and shared with users at the project level, so you can control who can edit your data and who can view your data. The storage itself follows the 3-2-1 storage strategy, so we have 3 copies of the data on 2 different types of media, with 1 copy far off-site, and the main high-speed copy is located in Canada on the University of Alberta campus. The annotation interface at its core is an interactive spectrogram like many of these platforms. So users can listen to the recording, view the spectrogram at the same time, and then draw boxes around annotations and provide data for those annotations. But we've recently upgraded the interface, and so it's now so much more. So now there's a tab that provides metadata about the recording, like time of day and weather conditions. You can view photos of the recording location if they're available. You can view the location over satellite imagery, add metadata about background noise. And we now have an AI overlay that provides predictions from both BirdNet and HawkEars, which is a Canada-specific classifier with user control over the score threshold. But you can also annotate in other ways. So you can use a completely human approach, like we just looked at. You can use what we would maybe call an AI in the loop approach, where you turn on that AI overlay. You could use a human in the loop approach, where you process your recordings with a custom classifier. You upload the detections, and you can use the verification interface to remove false positives. Or you can go full AI and just upload recordings, request processing through BirdNet and HawkEars, and then download the output straight into your analyses. Once you're done annotation, then we have publication and sharing. So WildTracks is aligned with both FAIR and CARE principles. And again, you have user-based role control at the organizational and at the project level, enabling you to control who can view and edit the data at both levels. There are varying levels of project publication status, ranging from all the way fully private, all the way to fully public. And there's also options for location visibility and buffering, say if you want to make your data public, but you want to protect sensitive location information. If your data are public, they're discoverable by other folks in the Data Discover portal, and then you can download them in tabular data either through the online interface, or we do have an R package called WildTracks. And so just to show, oh, there it goes. Just to show an example of how we at the ABMI use WildTracks to support end-to-end biodiversity monitoring and conservation. Oh, no, sorry, there's too many pictures. We use it to store and share data from our nearly 4,500 survey locations across Alberta. We use that privacy feature of buffering the locations to support long-term monitoring on private lands. The annotation options allow us a range of options from full human annotation when we're concerned about the influence of false positives in our analyses, through to full AI if we want to study things like phenology. And then, of course, WildTracks supports our end-to-end reproducible workflows, allowing us to download the data straight into the R. And so one of the key end products of these workflows are these large-scale species abundance maps, which we share with partners at the provincial government and industry to undertake land use planning and understand human disturbance in Alberta. Other collaborative applications that WildTracks supports is species density modeling at the scale of Canada, made possible by the data harmonization and standardization across all the projects in the WildTracks system. And then the data sharing and that data discover portal make other applications like reuse of data for impact assessment possible. And so the figure on the right here, you can see the accumulation of recordings in the WildTracks portal since the launch of the acoustic sensor in 2018. That's the gray line on the right. The red is actually camera trap data, which we also support in WildTracks. So there's been nearly 3 million recordings uploaded since 2018. And I think this collective effort really highlights the collaborative capacity of passive acoustic monitoring, especially when supported by robust data management systems. So with that, thanks to our partners who make WildTracks possible, and feel free to email us with questions.
Thank you, Ellie. So our last speaker is John Quinn from Furman University in the US. He will be presenting fine-scale acoustic monitoring grassland bird responses to agriculture disturbance.
Alrighty, so I'm the applied ecologist in the group and super grateful for all the technical work that everyone who has talked before has done. So I'm going to share the results of some of the work we've been doing for the last 5 years. Across the US, we're going to focus in on one particular area. And what I want to highlight is how the fine temporal scale of bioacoustic monitoring allows us to ask questions for conservation that we might either have glossed over before or have not been able to ask. I also have a couple other slides at the end because I wasn't sure how much time I would have at the end. I'll gloss over those quickly, but I think they make an important point. we're all here for conservation. My work primarily focuses on how do we bend that curve beyond protected areas, focusing on landscapes managed for forestry, sustainable agriculture, urban landscapes, so that sustainable production practices associated with the sustainable consumption that the choices that we make can create positive change. When we think about agricultural landscapes, I do most of my work in the Central Great Plains of the US. You drive across that landscape, it's corn and soybean over and over again. But there is a movement to start to diversify that. Maybe not going back to historical practices, but start to think about different systems, management ideas. There's lots of buzzwords out there: organic, sustainable, regenerative, but looking at individual practices and timing of those. Same with forest, same with urban landscapes. I want to talk today about work that we're doing in the Central Great Plains. The Central Great Plains is historically a mid-grass prairie, but in the 1800s, 1900s, heavily converted into an agricultural landscape. I always appreciate, and one question I have about bioacoustics broadly is, we describe these as natural landscapes, but Errol Ellis came with the city of anthromes. What do our anthromes sound like rather than other baselines? So this I would describe as a cropland anthrome, 90% agriculture. It has and continues to shift and becomes more intensive. So what have we been finding focused in these areas? On the first pass, we are finding a rich, let me take one step backwards since I have maybe a little more time. We deployed our ARUs across 35 farms in these landscapes. Each one is adjacent to a field. We didn't want to get plowed over by the tractor. And so we're detecting the birds on the edge and within the fields. From a comparison to our historical way we would collect that data, we are clearly capturing much more complexity and diversity in the landscape. So in this graph, each row is an individual bird, each column is a day, and then the size of the dot represents the number of times we detected a vocalization in the recording. We see things in a given year. Dick thistles are the large yellow blob. They're a very loud bird. They take up a lot of our recordings. Once they fade away, maybe we can pick up some other things. But you can see how sampling near continuously allows us to pick up species that we might not have known were used in those agricultural fields in a specific way. We've been sampling on these same 35 farms for 5 years now, and we're starting to see changes in phenology, perhaps changes in how species are using it in a migratory process, where some of the species nesting in Canada might be coming back to through Kansas using those fields. Particularly, maybe the farmer harvests a little bit later, or they leave the stubble, or they wait for different practices. We can also break that down by individual farm, which is each one of these columns represents, and looks at over the years the shifting communities as the farmers are adopting different practices. This project started in 2020. Farms were all kind of the baseline, and then each year, each farm is picking up a different suite of practices. So what we're working on right now is identifying which practices are having a positive outcome. A first look here is cover crops, if the farm is identifying cover crops or not. We can see species like the scissor-tailed flycatcher, the larkspur are responding positively to the cover crops. Others are not. What I'm interested in our next stage is to break this down by warm season, cool season cover crops, something that has been a good question, but it's hard to sample across sites because, one, not every farmer might put those cover crops in the same time, having to send a crew out to do replicated samples over space and time in each one of those. But if we just leave our sensors out there, over the years, we're able to answer questions more specifically. One thing I'm particularly excited about is we've asked each of the farmers who are participating in their project to tell us certain days when they're doing specific practices, whether it's harvest, putting down a cover crop. Here is pesticide application, and to say, are these specific practices on this day having an impact? And then how do the species recover or not? Across the suite here, we can see that most of the birds do decline when pesticides were applied. And this is a question we've asked before: is a farm, do they use pesticides or not in a binary? Now we can say, when did they apply pesticides and how did the species respond to it? The second thing we want to follow up on that is the recovery time. Do they recover 2 weeks later, 3 weeks later, or do they not recover at all? And then also looking at specific farm recommendations. Say farm A, you should delay your harvest a little longer. Farm B, you can go a little sooner based on the information we know about the birds that are present. I'll say briefly, we're also working with the Longleaf Alliance on biodiversity credits to say, can passive acoustic monitoring be a tool to have a verifiable high quality credit? I think some of the data processing that we heard about earlier is going to be really important for that in the long term. What we measure though, I think as a field, we need to push against richness as the measure used for biodiversity credits. We did a demo project at a single site that had 6 different age classes of longleaf management. And you can see on the bottom is measures of richness, not a lot of variability. If you look at the top, taking number of counts either on its own, which is on the right, or number of counts multiplied by a conservation score, gives us much more nuance or variability in how these practices are being implemented and whether or not, if a landowner is being paid for that practice, is it having the outcome that we want to see versus richness, which masks a lot of information. So I think when we think about credits, when we think about conservation, we think about research, we need to take advantage of that temporal data because it's there. And building projects around that is important, but you need good data management to get there. I think I did it without going all the way through. All right, I'm not going to hit questions on the next one. So I think with that, that's what I got.
Thank you to all our 7 speakers for great presentations. We now have, I think, 20 minutes for questions. So, yeah, Carly first.
Sort of, I think for both of you, what's the funding source behind these platforms? Because, obviously it costs a lot of money to store and manage these data. And I think figuring out sustainable funding streams for these types of archives and analysis platforms is coming out as one of the big kind of barriers. So curious how you have solved or are attempting to solve that problem.
So great question, Carly. I think with WildTracks, the important thing to remember is that it is 10 years old. And so what I presented today is the accumulation of a very small budget for 10 years. But our primary funding does come from our federal government because WildTracks was intended, has become a way for them to store and manage their own passive acoustic monitoring data. In Canada, the vast majority of the country is uninhabited and there's not a lot of roads. And so we've been learning to use passive acoustic monitoring for a decade. So that, our federal government has very strong storage and management needs. And that's how we've been able to put it together. But I think the important thing to remember is that it's really like one programmer over 10 years. And it really is the accumulation of time that has allowed us to build that program.
Okay. Okay, so for my project, actually the main funding is from my research institute. So we have an internal funding every year. So that can maintain the basic work associated with this project. So I can maintain the monitor project. And then I also apply for the grants, the grants for different topics. For example, we also studied how the impact of climate change on plantations. So we have some monitoring sites set up in tea plantations. So we using the different grant projects to expand our the network of the monitoring. So that's how we can monitoring.
Hi, Peter de Smet from IMBO. I have a similar question, but also to Nicolas. So for PAMFlow, WildTracks, and Asian Soundscape, how open are you to us sending researchers your way to use your platform for managing their data rather than developing their own platform?
our idea is for our intent is for other researchers to use it for managing their own data, because as I stated at the beginning of the presentations, I find that we find that many of the time, since passive acoustic monitoring computational tools are not developed in an open way, teams are forced to develop their own software, and we want to finish that. We want people to use our software. Our repository is actually open, and it is open to pull requests. So pretty much.
I want to clarify the question. So it's about how to whether we can encourage other people to use this platform, something like, oh, yeah, yeah, okay, okay, yeah. At the beginning, when we established this data portal and also actually this monitoring network, we hope that the other researchers can also upload their data to this data portal, so we can have a platform to integrate the data from different projects. But later on, we found there is a big challenge. It's about the funding, because we maintain this current networks already spent a lot of money. So currently we are hesitate to encourage other people to put their data to our network. One thing is especially for the storage. So currently our data is already 600 terabytes, and it costs a lot of money just for storage. So so that's a limitation or the reality. So we face it right now.
Yeah, it's a great question. So I think like we do already have users from across the globe in WildTracks. The majority of our user base is in Canada. I think, you know, we're certainly not ready, and frankly, I'm not sure it would be appropriate for WildTracks to be a completely global one-stop shop. I think modularity between regional systems is a better solution. But if folks are looking for a place to put data, you're welcome to come join us at WildTracks. There is, because we offer that 3-2-1 storage strategy, like if you're uploading, I think it's more than a terabyte, then we do have a cost recovery strategy for data storage, because it costs money to maintain the servers at the University of Alberta, but you know, then it circumvents you having to have a whole bunch of hard drives in your office. So yeah, the short answer is like, come join us, but the long answer is we should work towards supporting regions.
Yeah, my question is for you actually on wild tracks, because I heard you mentioning abundances, and I was wondering how you estimate abundances in wild tracks.
Yeah, so full disclosure, I'm actually the quantitative person. I'm not the developer or the manager. So because a lot of the data that we've collected in Canada is stereo, and it's all actually like those 3 million recordings that I mentioned are primarily human annotated. We haven't actually used AI that much. And so how we're getting to count is because of those stereo recordings, it's essentially like doing a point count. We annotate to the first detection of every individual of every species. So we have like hundreds of thousands of recordings in WildTracks that have count data associated with them. And like, if anyone wants to work on, you know, methods for getting to individual ID or to count through call rate, like we're sitting on a pretty great data set. So, you know, please come talk. But yeah, I mean, I think other than missing the visual cues, it's very similar to doing a point count. And we actually have a paper coming out in ornithological applications soon, comparing some components of the detection process between point count data, like in-person point count data versus what I think we would probably call this like a recording point count, what we're doing. And there are some differences, and I think you actually sometimes get better data doing human annotation of the recordings than you do with an in-person point count, because you can stop and pause the recording. Anyway, I could talk a long time about this, so I'll stop.
Great. There are individual ID efforts from Bioacoustics that are like in the last 2 years from one of the co-organizers, Justin Kitz's lab, and Sarah Beery's lab at MIT. So watch this place.
Hi, everyone. I've rather a question about the identification skills and the accuracy about it nowadays. Since there are different tools, Merlin, BirdNet, etc. I've seen in, for instance, the presentation by the Taiwanese researcher that you're using clips. Is it automated, or do you use fixed duration clips, about 5 or 3 seconds, or is it human interpreted?
Yeah, so we use the two AI based approach. So one is BirdNet. So for the BirdNet is 3 seconds as a clip, right? So it's identify the bird species within the 3 second period. So, but another tool we use is Silic. It's developed in our collaborators in Taiwan. So it's focused on the vertebrate species in Taiwan. And in that tools is identify the it is an objective oriented approach. So it will mark the locations of the codes within spectrograms for each detection. And so in that application, so there is no so it's we can do that for the entire recordings. Currently, our recording is 1 minute. So all of our recording is standardized as 1 minute. And so, but for BirdNet is identification for say.
As regarding the identifications, is it species specific that you look into clips based on some experiences in Europe where some song thrushes, for instance, vary very much every 3 seconds, different songs. So the percentage of ID is very low compared to one repetitive song by species.
Yeah, I understand that, and but currently, I mean, for the BirdNet, it's the limitation set by the program, right? So I don't know. Probably other people can know. Other people knows how to release these limitations. But as I mentioned, for our tools we use, there is no this kind of limitation. But for BirdNet, we also try different overlaps. So it's some kind can avoid or reduce the effects of that.
I just think there's a lot of different models out there that use different non-overlapping time window lengths. So Birch is using 5 seconds. Different groups are making their own models, or transfer learning off of those foundation models. And I think the performance, if you're asking about performance by class, like by species, that varies by where you're using the model. So, a song thrush would vary in Northern England versus, Southern England. So there's a lot there that still needs people in the loop and people thinking when they're deploying these things, and how it is that you're interpreting these machine predictions. Happy to talk afterwards.
So thanks for the incredible talks. This is again for the platform folks. So since you use AI models, BirdNet is one of them, which is kind of focused on birds, but that's okay. There's lots of birds to detect. Would you be open, because this also ties into the cost question, to partnering with people who have compute money and who want to develop models that are across species, more general, that can handle different sampling rates, for example? Because underwater you have different sampling rate requirements. Overwater, different species are at different frequency bands. So would your platform be open to communicating with models outside the servers, and that would also relieve you from the cost of compute?
Yeah, it's a great question. I mean, I'm not the manager of the platform, so I can't, speak on behalf of Christina Kopp is the manager of an information center at ABMI. I think incorporating other models is something we've been talking about for a while. I mean, especially sort of are building custom classifiers to get to things like individual ID every day now. So I think it's something that's been on our priority list for a while, and yeah, would love to talk more about it, but we're not there yet, I guess is the short answer.
So yeah, before continue taking questions, we had a question that we wanted to do to all of the speakers, and I think we were talking a lot about this, but given the diversity of this panel and the different regions you all represent, what do you think is most needed in your context to ensure that bioacoustic doesn't remain isolated in projects or within individual organizations, but are shared and accessible and connected to global frameworks like GBIF or OBIS? And also maybe, Sam, you can speak about the hosted data portals on GBIF if you want a little bit, or others.
Okay, thank you. I missed a bit this first part, but you want me to talk about the hosted data portal? Yeah, okay. So, yeah. So now on GBIF, it's a bit difficult to find specific like camera trap data or acoustic data. And the idea is that there would be a hosted data portal. Peter will create it or is organizing it. And it's based on GBIF, and all the data sets that are in ChemTrapDP format can be found on that portal, and there will be more specific filters to search in that data. So at this point, it's only camera trap data, but the idea is that it would be also be for acoustic data, and that you can filter on which type of media it is. Does this answer the question?
Other speaker wants to add?
So basically, in the follow-up to this horizon scan, we're also thinking about what does this community of practice need, and how do we move forward together? Like, we just had the launch of MoveBond last night. That community of practice made sense for that community. And so I guess in your reflections for our speakers, if you can also touch on like what do you think is needed, not just in terms of unlocking biodiversity or bioacoustics data globally, but like as a community, how do we collectively move forward? Peter.
Yeah, one of the things, so I've been working in the camera trap community, and there's a number of big systems, Wildlife Insights, Trapper, Agouti, where people can just upload their data for free. These systems are infrastructures that are maintained through different projects, and I'm far less aware of the landscape in bioacoustics, but I've now heard of 3 platforms that are semi-open, but also don't want to overload the team that is maintaining this. So I think one of the things that is maybe missing in the bioacoustics community is these bigger platforms and funding for that infrastructure, so people don't have to reinvent the wheel, don't have to map their data to CamTabP manually, but that they can just upload it and it's more seamless to manage data, publish data. So yeah. Yeah, exactly.
Yeah, and I think I think that's a really good answer. And I just think you know, maybe a targeted community of practice or perhaps a steering committee that facilitates modularity between the platforms. I mean, I think CamDP is a great place to start, but there's so much richness in the data. And I think there's a lot of lessons learned in building some of these platforms that I think there'd be a lot of value in that knowledge sharing and collaboration.
I'll also say from as a faculty member, training the next generation of ecologists to understand large quantitative data sets. I think a lot of people get into the field because they want to run around the woods and, you know, hug trees. And then they hit their first job and they're like, oh, wait, I have to know how to manage a database and I have no idea on those different practices. And so I think some sort of education for the next generation so that at meetings like this, it's a diverse mix of people.
So with Nicolas and the team at the Humboldt Institute, so just to clarify, it's not a platform that we have. It's just a workflow that anyone can use and can adapt to their own needs. So that's what we put to the community so others can build upon, because that's what happens. Everything is moving very fast. And if you build maybe a platform, so it's very cool, but you need funds to maintain it and then to adapt it to everything that's moving so fast. So we said, okay, let's build, so in this level of abstraction where you have like functions, lots of different functions in Python, but you need a workflow. We built something a little bit closer to a platform, so a backend where people can build a front end or adapt it. However, so that's part of the little by little solution that we find, I think, can be useful. So we can have nodes. So I really agree here that I see this more as having different nodes that can interconnect data and share it and then ship to GBI.
Thank you very much. We are on time, so there's much more to discuss, and we hope we can continue discussing this in our International Conservation Technology Conference in Lima, Peru. So now is a call for session open until November 15. So maybe we can think about some sessions to continue discussing how to advance these priorities. So thank you so much to all of you.
Okay, for let's start into and then season going. See, está aquí equipo. Vas a estar contigo, ¿cierto? Igualmente. Yeah.